Okay, so when AI hits the headlines, you usually hear about, text generation or maybe cool
Right,
images.
the flashy stuff.
Exactly. But the really big shift, the seismic one, it's happening kind of behind the scenes, where the physical world meets the virtual.
Hmm.
AI isn't just running simulations anymore. It's actually designing physical molecules. It's automating entire lab procedures. It's fundamentally rewriting the R&D playbook, especially in biomedicine.
Yeah, absolutely. And we're talking about validated, tangible results here.
That's the key, tangible.
And, this deep dive, it's really your fast track into that new reality. We've gathered a stack of, well, frankly, breakthrough papers. They show how specialized AI is creating this continuous, almost autonomous R&D loop.
Autonomous. Wow.
Yeah, we're talking LLM-guided discovery, these amazing generative models like Boltzgen we'll get into, and AI that literally writes the step-by-step instructions for lab robots.
Hmm.
So our mission today is really to unpack the most surprising stuff and show where human expertise, scientific knowledge, meets machine speed.
Right. And we're aiming this at an audience that gets that the future of drug discovery isn't just about being faster. It's about being deeply interdisciplinary. So let's unpack this. Starting with a concept that honestly still sounds a bit like science fiction, the virtual lab.
Okay.
So picture this, an AI research team. A human researcher guides a team of specialized LLM scientist agents.
Like little digital experts.
Exactly. And they're all supervised by an LLM principal investigator, a sort of AI boss.
It's kind of the ultimate collaboration, isn't it? To build something really complex, let's say a new nanobody,
Hmm.
these LLM agents, they have to reason across immunology, protein folding, and machine learning,
Yeah.
all at the same time.
That's the mind-bending part. So the human gives the strategic direction, the what-if questions.
Precisely. But the machine handles the incredibly complex job of synthesizing all that interdisciplinary knowledge and, doing the heavy lifting of designing the molecule.
And the speed. You mentioned the speed. That's what floors me. So this virtual lab, they were tasked with designing nanobodies against recent SARS-CoV-2 variants, specifically JN.1.1 and KP.5.3. These are tough moving targets.
Very tricky.
Yeah. They started with just 4 known nanobodies as a reference point, and the AI team, it rapidly came up with 92 nanobody candidates.
92. I mean, just think about that. That's like running a massive high-throughput screening operation, but virtually, instantly.
But was it just theory? Did it work in the real world?
That's the critical part. This wasn't just spitting out sequences. These candidates were then actually made and tested by human researchers in a wet lab.
Mm.
And get this, the agents created one mutant, NB21, that didn't just bind better to the JN.1 target.
Okay.
It also gained the ability to bind to the KP.3 target. That's huge.
Wow, so it generalized across variants.
Exactly. It proves the system can successfully adapt these really complex methods to emergent, real-world scientific problems.
Mhm.
It's like having an instant cross-functional expert panel on call.
Okay, that success, though, it brings up a huge practical question. If the AI can design 92 molecules that fast, how do you actually test all 92 efficiently and accurately in a real lab?
Right.
Because the bottleneck for these self-driving labs has always been reproducibility, hasn't it? Especially with, well, how messy scientific literature can be.
You hit the nail on the head.
Yeah.
Exactly. The way humans write science papers,
Yeah.
even for something complex like, say, Fischer-Tropsch synthesis,
Yeah.
it's great for understanding the why.
Sure, the narrative helps us learn.
But it's terrible for automation. Procedures are scattered all over the place. It's written in free text and often lacks the super precise detail a machine needs to do the exact same thing every single time.
Right, you can't have ambiguity for a robot. So we need to translate that human narrative mess into a robot-readable instruction manual.
Precisely. And that's where this KG-LLM framework comes in. It pairs a domain knowledge graph, a KG, with a large language model.
Okay, KG-LLM, how does that work?
So the KG, the knowledge graph, that's the key to consistency. Think of an LLM
Yeah.
like a brilliant, super talkative assistant who sometimes, makes stuff up, hallucinates.
Right, we've all seen that.
If you just ask the LLM straight up to write a protocol, it might invent steps or conditions. The knowledge graph steps in first. It cleans up all that unstructured data from the papers, reagents, equipment, actions, temperatures, and turns them into structured, traceable triples, like subject-verb-object facts.
Ah, okay. So the KG acts like the factual memory, the ground truth.
Exactly. It provides the guardrails. Then the LLM uses this structured, factual input, guided by an outline an expert human wrote, to generate a clear, logical, step-by-step protocol. And because every single instruction can be traced back to the original source text through the KG, You basically eliminate the LLM's tendency to just invent details.
And does it actually work? I mean, in
The
practice.
proof is in the pudding, as they say. This KG-LLM approach, it achieved an F1 score of 92.31% for factual reproduction. That's really high.
92%. Okay.
Yeah. And maybe even more importantly, it scored way higher on things like coherence, consistency, and relevance compared to older methods that just relied on detailed prompts.
So it's the difference between trying to build IKEA furniture using random forum posts versus having that perfect, structured, robot-readable instruction manual.
That's a great analogy.
Okay, now let's pivot a bit. From designing specific nanobodies to the frontier of creating universal molecules. We need models that don't just solve one problem, right, but can invent whole new classes of molecules.
Yeah, the generative challenge.
Enter Boltzgen. You mentioned it earlier. This is described as an all-atom generative model for high-affinity molecules, proteins, peptides, nanobodies, and it unifies design and structure prediction.
That's right. And what's really fascinating here is how Boltzgen tackles the Achilles heel of a lot of generative AI generalization.
The generalization challenge, meaning does it actually learn or just memorize?
Exactly. So many models look good on benchmarks only because their test data is secretly really similar to their training data. They're memorizing patterns, not truly generalizing principles.
So how did the Boltzgen team test its real generalization power? Avoid that trap.
They were very explicit about it. They validated Boltzgen on what they called novel targets. These were 9 proteins that had very low similarity, less than 30% sequence identity, to any protein with a known structure bound to something in the big database, the PDB.
Less than 30%. So these really look different from anything the model had likely seen during training.
Right. If you don't look like the training examples, you are a tough test for the model.
Okay, the moment of truth. What happened with these 9 challenging, truly novel targets?
Get this. Boltzgen achieved nanomolar affinity binders.
Nanomolar. That's incredibly tight binding.
Extremely tight for 66% of them. 6 out of the 9 novel targets. And that success rate held up for both nanobody designs and general protein designs.
6 out of 9 on totally unseen targets with nanomolar affinity. That's pretty amazing.
It's a huge statement. It suggests the model is genuinely learning some of the underlying physics of how proteins interact, not just pattern matching.
And it wasn't just one type of molecule, right? The diversity was broad.
Oh yeah, remarkably broad. They designed proteins to bind complex things like bioactive peptides into lecithin, melittin. They generated these disulfide-bonded cyclic peptides, got 14 binders out of 28, tested against a tricky dimer target.
Wait, did you say it even worked for small molecules, like drugs?
Yes, pushing the boundaries there too.
Ah.
It successfully designed proteins that bind to common small molecule drugs, like rucaparib and even a rhodamine derivative. The affinities were weaker, maybe micromolar range, but definitely detectable.
Okay, this is impressive stuff, but here's where, for me, it gets really interesting. The part that signals a major shift in how R&D might work.
Ah, I think I know where you're going.
Boltzgen wasn't kept locked up, was it?
Nope. The creators released the entire package, the model weights, the training code, the inference code, all of it, open source, MIT license.
That's huge. State-of-the-art biomolecular design capability just dropped into the public domain, available to any academic lab, any startup.
It really democratizes the ability to start creating new potential therapeutics. It lowers the barrier significantly.
Okay, so we have AI designing molecules, AI writing lab protocols.
Right. Now let's move on from design to the analysis side. Because even with the best generative models, figuring out the exact 3D structure of really dynamic or unusual proteins, that's still one of the hardest problems in biology.
We need ultra precision there.
Exactly. And that brings us to a technique called protein test time training, or protein TTT.
Protein TTT. Okay, this sounds a bit counterintuitive. You're saying it uses self-supervised learning to customize these big protein language models like ESM-Fold.
Right. Models trained on billions of sequences.
But it customizes them on just a single target protein right at the moment you're trying to predict its structure. Why retrain on one data point after all that initial training?
It seems weird, right? But think about it like this. Even the best models can get confused by sequences that are really different from their training data. In language models, there's this metric called perplexity, basically, how surprised or confused the model is by the input. If a protein sequence is very unusual, the model's perplexity is high, and its first guess at the structure is often poor.
Okay.
Protein TTT works by forcing the model, right there at test time, to minimize its perplexity for that specific sequence. It makes the model quickly adapt its internal understanding just for that one unique protein.
So it's like a quick, focused cram session for the AI right before the test.
Kind of, yeah. And that adjustment, which sounds maybe marginal, result is anything but
How big a difference does it make?
transformative. In one example they showed, protein TDT took an initial prediction that was really low quality, a TM score of 0.29,
Which means,
which basically means it was a useless smudge, completely wrong-fold.
got it, useless smudge.
And protein TTT refined that smudge into a nearly perfectly folded protein, hitting a TM score of 0.92.
Wow. From 0.29 to 0.92. That's not just tweaking. That's the difference between total failure and pinpoint success in structural biology.
Exactly. A massive improvement for those really challenging cases.
And we're seeing a similar push for precision with dynamic systems, right? These are super important drug targets.
Absolutely. Things like membrane proteins, channels, transporters, they move. They change shape to function.
So one study combined generative AI specifically using AlphaFold 2 in a clever way, stochastic subsampling with simulations, density-guided molecular dynamics.
Right, using cryo-EM data.
To model all the different shapes, the alternative conformational states these proteins can adopt.
This is so vital, because if you only model one static snapshot, you might completely miss the actual site where a drug needs to bind, a site that only appears when the protein shifts its shape.
So they tested this on a really tough case, the ASCT2 transporter, which undergoes huge rearrangements.
Yeah, a classic example of major conformational change, and the results were stark. The AI-generated ensemble approach modeling all those different states produced a final model with an accuracy of 3.65 angstrom C alpha RMSD.
Okay, 3.65 or MSD. Translate that for us. RMSD means.
Root mean square deviation. Lower is better.
Yeah.
It basically measures, on average, how far the atoms in the predicted model are from their true positions in the experimentally determined structure. 3.65 angstroms is incredibly good, close to atomic resolution for a complex like this.
And what if they hadn't used the AI ensemble method? If they just started from a single known structure?
If they started from just one known experimental structure, the accuracy they got was 10.16 angstroms RMSD.
10 angstroms compared to 3.65.
10 angstroms is, well, it's essentially a failed model at that point. It shows that for these really complex dynamic proteins, you need that AI ensemble approach to capture the biologically relevant structures. The single snapshot just doesn't cut it.
Okay, so we've seen AI designing molecules, automating labs, predicting structures with incredible precision. But amidst all the success rushing in, we absolutely have to pause. Take a breath.
Time for some critical thinking.
Yeah, because our sources are very clear. The AI hype needs serious quality control. We need to talk about the data trap.
Ah, yes, the data trap. The hidden problem of memorization versus actual learning. It turned out that the initially stellar performance numbers for many deep learning models, especially in things like drug docking or binding prediction, well, they were heavily driven by data leakage.
Data leakage, meaning?
Meaning the test data used to evaluate the models was way too similar to the data the models were trained on. One analysis found that a shocking 49%, almost half, of the complexes in common benchmarks like CASF were structurally very similar to complexes in the PDB-bind training set.
Half. So the models weren't really generalizing to new things. They were just acing a test on stuff they'd basically already seen.
Pretty much. They were acing a memorization test, not a generalization test.
That really undermines the claimed success rates of potentially years of research in that area. How did researchers even fix that? How do you ensure a fair test?
By building better data sets, specifically by creating the PDB-bind clean split dataset. They used a clever structure-based clustering algorithm to go through the data and strictly remove any complex from the test set that had high structural similarity to anything in the training set.
Forcing the models to actually generalize.
Exactly, forcing them to predict interactions for genuinely unseen protein-ligand pairs. And surprise, surprise, once this data bias was removed, the true generalization performance of many existing models was shown to be much, much lower than previously reported.
Wow. It just proves how absolutely essential that kind of data vigilance is. You need clean data for real progress.
Absolutely crucial.
And this whole issue of specialized versus generalist AI also came up starkly in drug target discovery, right? There's this framework, Target Bench 1.0.
Yes, Target Bench. It was built specifically to evaluate different AI models on how well they identify promising drug targets, targets that are actually suitable for moving into clinical development.
So the specialized versus generalist showdown, what happened?
Well, the results were pretty definitive. There was a specialized model, Target Pro. It's a supervised learning model, explicitly trained on multiple types of biological data relevant to target discovery.
Okay.
Target Pro achieved a 71.6% retrieval rate for known clinical targets. Pretty impressive.
71%. And the general purpose LLMs, the ones everyone talks about.
They didn't fare nearly as well. Their retrieval rates ranged only from about 15% up to maybe 40%.
15 to 40%, compared to over 70. The specialized tool absolutely crushed the generalists in this specific task.
Completely.
But wait, that goes against the grain a bit, doesn't it? We keep hearing LLMs are going to take over everything. Why did the specialized supervised model dominate so thoroughly here?
Because discovering good drug targets isn't just about processing text. It requires integrating very specific, structured biological data, understanding complex biological pathways, assessing clinical relevance, things that require domain expertise that general LLMs just haven't mastered yet, at least for this task. And it wasn't just about finding known targets. Target Pros prioritized novel targets. The potentially new drug targets were demonstrably higher quality.
But that's so.
Well, 95.7% of Target Pro's top novel suggestions had an available 3D structure, which is critical for starting drug design. And 46% had potential for drug repurposing. The LLMs just couldn't match those numbers on metrics that are really critical for actually advancing a target towards the clinic.
So for these really data-intensive, specialized scientific applications, the tailored, supervised models still have a significant edge.
At least for now, definitely. It underscores their value.
Okay, let's try to pull this all together.
Sure. So to quickly summarize the sort of 3 layers of AI advancement we've really explored today. First, you have generative AI, like Boltzgen, that's creating and designing completely novel molecules at incredible speed.
Right, the invention engine.
Second, you've got predictive AI, supercharged by techniques like protein TTT or those ensemble methods for cryo-EM. These are solving the really tough structural and dynamic problems with much higher precision.
Tackling the hard analysis.
And third, there's systemic AI, the KG-LLM approach, which is starting to automate the entire lab workflow, making experiments more reproducible and scalable.
Putting it all together in the lab.
But, and this is the crucial but, the success of all 3 of these pillars depends fundamentally on that critical, often human-led effort to ensure data quality. The PDB-bind clean split story proved that. Vigilance is key.
It really feels like the era we're entering isn't just about using AI as a tool to speed up what we already do. It's becoming more about collaborating with AI on the scientific reasoning itself.
That's a great way to put it.
I mean, if these LLM agents can rapidly learn and adapt complex methods from immunology, protein folding, machine learning, all at once to design new nanobodies, the main challenge for human researchers starts to shift, doesn't it?
Absolutely. Less about the manual grunt work, maybe.
Yeah, less about pipetting or even just collecting data, and far more about providing the high-level feedback, the critical judgment, the strategic direction needed to steer these incredibly powerful interdisciplinary virtual teams.
Guiding the AI collaborator.
Exactly. So as this autonomous R&D loop starts spinning faster and faster, here's the final thought for you, our listeners. What high-level strategic feedback would you give that virtual lab team for their next big scientific project? Because figuring out how to provide that guidance, that's the strategic shift we are all going to have to navigate very, very soon.